All right, guys, this is my third attempt at recording this as the programs keep crashing on me. So third time's a try, right? So we're in the section of our notes that talks about controlling gene expression. And so really the connection to what we've been doing, we're talking about making a protein, right? Transcription and translation create a protein. Well, that process needs to be controlled. It needs to be regulated. First, it is not terribly energy efficient for a cell to constantly be cranking out proteins if those proteins aren't really needed. It's like a waste of energy. Second, I would say not all cells need to make all types of proteins that are encoded in your genome. And so we'll start first with the why in the world do we need to control protein synthesis? Why do we need to control our ability to express a gene, which at its core is making a protein? Now, this goes back to something that we've talked about in a previous unit when we discussed mitosis. So all of your cells in your body, they have the same DNA inside of their nucleus. So it doesn't matter which cell you sample out, all of your cells essentially have the same DNA in their core. And that is because you started as a single cell. When sperm met egg, you started as a single cell, and that cell was copied and copied and copied by the process of mitosis, which we know is an identical copying process. So every cell in your body has the same DNA inside of it, but not all cells are the same, right? So think about nerve cells. Nerve cells look and function very differently than skin cells. So even though they have the same DNA in their nucleus, there is a control mechanism on what parts of the DNA are going to be expressed in the neuron compared to the skin cell. And so that control process is important, number one, in first creating those different cell types when we were an embryo, creating those unique cell types, but also in creating the proteins that those cell types need to continue to live and grow. So all of your cells have the same DNA inside of them, but your cells are different. All of your cells are different. And really, at its heart, that means that not all of the DNA inside each kind of cell needs to be expressed at the same time. And so we need to control that process. Now, we're going to watch a quick section of this video. It's not even the full video. If your tendency is to fast forward through these videos, please, 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 please watch this time, because it's a topic that really is covered in this unit, but we don't have a ton of time to dedicate towards it. So we're going to let the video teach us a few things. So check out this.
We've mentioned a lot about specialized cells. Specialized plant cells, specialized animal cells, so many kinds of specialized cells, it's going to get a bit crowded here. But have you ever wondered, how do they get specialized? How does a neuron or a muscle cell in your body have the structure and function they have? I mean, can you imagine if they had to switch jobs for a day? That wouldn't go so well. They're so specialized for the function they perform. Well, this video is going to talk about how cells differentiate into other cells, which basically means how cells become specialized. Remember that many multicellular organisms, like a plant or you, well, these organisms come from a fertilized egg cell. Let's take a look at a human fertilized egg, otherwise known as a zygote. Well, that zygote divides to make more cells. And more cells? Oh, look, it's a morula. And more cells? Oh, look, it's a blastocyst now. You know, the problem is, if the cells just keep dividing, if you remember from our mitosis video, that makes identical cells. Well, that's great for growth, and so dividing is definitely going to happen, but that alone is not going to result in different specialized cells with different specialized functions. There's something else that will be happening for that. So let's take a look at that in a bit of detail. We're going to pause here, the blastocyst. Notice it has stem cells, and these stem cells, they're amazing. See, they're not differentiated yet. They're not specialized. They're like blank slates. They don't have a special structure. They don't have a special job. They can become any type of body cell. Now, a reminder about body cells in your body. They all, with a few exceptions, contain all of your DNA. So neurons and muscle cells in your body, they don't have different DNA. They use different parts of your DNA. Genes are regulated, which means the genes can be turned on and off. It's important to understand because that's a big part of how these stem cells are going to specialize. Stem cells will activate certain genes in the process of differentiating into different types of cells. Transcription factors are major key players here. They're typically, but not always, proteins, and they determine which areas of the DNA code will get transcribed into mRNA. Now, that mRNA can eventually be used to make specific proteins that can impact what a cell is going to look like and what a cell is going to do. That means transcription factors have a major role in determining which genes are expressed in a cell. Because a cell that's going to become a skin cell is going to have different areas of genes expressed than a cell that's going to become a stomach cell. There are both internal and external cues for stem cells, which can involve these transcription factors. Examples? Okay, an example of an internal cue could be transcription factors present in the cytoplasm of the original starting zygote cell, which will eventually be present in the cells that come from it. Now, the specific location of the stem cell within the developing embryo can matter because the transcription factors available in different areas of the developing embryo can differ in quantity and type, which could impact what a stem cell differentiates into. External cues could involve cell signaling from other cells next to it. External cues can even be environmental effects like temperature. There's still a lot of research in this area, and we can't wait to see what scientists discover about this in the next decade. So,
all right, so I'm going to pause the video there. So one thing that we saw is controlling gene expression is really what gives us all of our unique cell types as a developing embryo. So different parts of the gene, different parts of the DNA, different genes are transcribed and translated, and that produces different cell types and gives us all these unique cell types. Oh dear, hold on. Cruise on to the next slide. So continuing this discussion of why controlling gene expression is what gives us all of those different cell types in the first place, right? So the first couple of cells that we are as we're developing are stem cells. They need some programming. And so the sort of creation of these proteins within those stem cells cause them to differentiate, to change the shape and then become specialized for a particular function. So you've seen these words before, differentiation and specialization, back in our cells unit. We have talked about how differentiation is really a process of changing your shape or your structure so that you can specialize in a particular task or function. So let's cruise into how do we control it. So why do we control it? Because cell types are different. We control it to get those cell types in the first place, and then we control it to produce the proteins unique to those cell types moving forward. How we control it requires a reflection back on transcription and translation. So there's nothing to write down in this moment. Just look at the screen for just a second. Let me make a connection for you. So this is the central dogma. Remember the first thing that we wrote down in our notes packet, the idea that DNA is converted or copied into the form of mRNA, and that's called transcription. And then mRNA is read and turned into a protein, or a protein is created. That's called translation. So when we talk about how do we control this process, we're going to break it down into control mechanisms on the front end of transcription, pre-transcription control mechanisms on the back end of transcription, post-transcription control, and then control mechanisms on the back end of post-translation. And so you could even think about pre-transcription control is going to be targeted at the actual DNA. Post-transcription control is going to be targeting the mRNA, and post-translation control is going to be targeting the protein. So let's dig into it in that order. So pre-transcription modifications. This is essentially impacting the DNA in some way that ultimately affects whether a protein is made or not. And you need a quick reminder of a histone. So you have this diagram here in your notes. Remember that histones are the sticky proteins that DNA will wind itself around to condense into chromatin and further condense into a chromosome. So in a minute, when we talk about modification, pre-transcription modifications, we're going to talk about adding some things, some little chemical groups to these histone proteins that will ultimately affect whether the DNA condenses or not. As you write this down, check out this animation of a histone real quick.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin.
Okay, so the purple things were the histones, and the DNA was wrapping around the histones, and it would condense itself into chromatin and further into chromosomes. So now that we know what a histone is, let's talk about methylating histones. So histone methylation is one way that we can control expressing that region of DNA where the histones exist, which is controlling gene expression. So what are we going to do? If you add methyl groups, which is a chemical molecule made of carbon and hydrogen, CH3, if you add methyl groups to those histone proteins, you have methylated the histones. And so here you can see the green, those are the histones, and these little red dots are the methyl groups. So if I put methyl groups on the histones, notice what it does. It pulls those histones together. And what that is doing is tightly condensing our yellow strands up here, which are DNA. So this is DNA that is open and accessible and could be read and turned into messenger RNA. This is DNA that's really tightly condensed, and it's hard to get in and read that DNA. So if I add the methyl groups, it causes DNA to condense, and you can't actually read it. You can't transcribe it, which ultimately decreases protein synthesis. If I remove those methyl groups, the DNA opens back up. So adding and removing methyl groups to histone proteins can either condense the DNA and make it difficult to read, or open up the DNA and make it easier to read. Adding the methyl groups condenses. Removing the methyl groups opens it back up. There is another chemical group called an acetyl group. Acetyl groups can be added to histones as well. So if I add acetyl groups, that acetyl groups kind of do the opposite. So adding acetyl groups to the histones where the DNA exists will expand that DNA and open it up and make it easier to read or transcribe, which will increase our protein synthesis. So one way of controlling gene expression at the pre-transcription level is to add and remove methyl and acetyl groups. The next sort of control mechanism that is pre-transcription involves activator transcription factors. So let's talk about ATF, or a transcription factor. It's really a fancy name for a protein. And that protein, if it is an activator transcription factor, it actually binds to the promoter region and helps RNA polybind. So it's like a helper, and you can see that here. Here is an activator transcription factor that is binding, and that binding allows this big gray RNA polymerase to bind and start the process of transcription. So activator transcription factors are helpers. They will start the process of transcription. There are also repressor transcription factors that do the opposite. So if a repressor sits down on the promoter region, it actually blocks RNA poly from binding, and you cannot transcribe that section of DNA. So there are two categories of transcription factors: activators and repressors. They will either help kickstart the process of protein synthesis, or they will prevent transcription from occurring and ultimately a protein from being created. Here is that repressor. So repressor protein binding to the same spot prevents the big guy here, RNA poly, from attaching and transcribing, creating some messenger RNA. The last type of pre-transcriptional control of making a protein is an enhancer region. And so we've actually seen enhancer regions when we drew out the basic structure or anatomy of a gene. Enhancer regions are additional regions of DNA that are typically located what is called upstream from the gene that needs to be coded. And so upstream just means in front of. These are the regions of DNA that sit in front of the promoter. And if they exist, and a protein binds to the enhancer region, it actually facilitates the process of transcription. It makes RNA poly bind to the real promoter region quickly, and transcription will occur and run pretty quickly as a result. So enhancers will boost gene expression. Next sort of target of controlling gene expression would be post-transcription control. So this is going to be controlling the messenger RNA that has been created. And so there are two things here that are going to control the mRNA and ultimately control the pathway of gene expression. So the first is called RNAi. These are RNA interference molecules. That's what the little I stands for. And they basically will degrade messenger RNA as it is leaving the nucleus and headed to the ribosome. So think about one way to control gene expression is to just destroy the messenger RNA. Then it won't land on a protein, and it won't land on a ribosome, and it won't make a protein. So RNAis do that. They degrade the messenger RNA so that it cannot interact with the ribosome and make a protein. There are two primary types of RNAi molecules. One is called an siRNA. Those work like up here in the blue diagram is showing an siRNA molecule, and that chops up your messenger RNA. The other is called miRNA, microRNA, and microRNA actually binds to the messenger RNA, which prevents it from entering the ribosome. And so it can't actually be read on the ribosome if it can't get into the ribosome. The last post-transcriptional control is splicing. And so there's not a ton to say here because we already know what splicing is, but I would just toss out to you that if you do splicing correctly, you're going to promote PS or protein synthesis. If you don't splice correctly, or if you splice slowly, then you're going to lower or slow your rate of protein synthesis. So controlling splicing could also be a control mechanism for gene expression. Again, focused on the messenger RNA, so it is a post-transcription control. The last type of control is a post-translation control. So this is a control mechanism that targets the actual protein that has been created in the process of protein synthesis. And so what I'll present to you today as an example of post-translation control is the interaction between something called ubiquitin and a proteasome complex. So first, let's go with ubiquitin. Ubiquitin is a protein, and here it is. And it is a tag that when added to another protein acts almost like a death tag. And so if you as a protein, if you've been created as a protein and you're tagged with ubiquitin, you basically are tagged for destruction. And so enter the proteasome. That is the protein complex that is going to destroy whatever ubiquitin is attached to. So if we make a protein, if we get all the way through transcription and translation, and we don't actually want or need that protein, tag it with ubiquitin, and the proteasome will come and destroy it. So that's an example of a post-translation. We made the protein, but maybe we don't want to express it. Tag it with ubiquitin, and it will be destroyed. All right, when you are done with this section, you are done with the control mechanisms in a eukaryote. So everything that we just talked about, that is in a eukaryotic cell, cells that are animals, plants, fungus, or protists. Prokaryotes do it different, and that is the topic of our next lecture that you'll watch on your next at-home day.